So I'm going to kind of do this as a lightning talk. So please ask questions or catch me later if I breeze past something too fast. So the scope of the whole system is genomic data. And for those of you who don't know, genomic data is mainly information about your DNA. And your DNA is this long string of 3 billion molecules. And each of those molecules, the names of them begin with an A, C, T, or G. So you can represent it as a 3 billion character string of A, Cs, Ts, and Gs. And the scope of the data that we're interested in is anything about any particular spot on that string. So we might want to say something like, in a particular person, we saw a T somewhere when we expected an A, and that's a mutation. And maybe that means that they're going to get sick, or maybe that means that a particular medicine won't work for that person. And DNA is at the center of a very, very complex interaction diagram for different kinds of data. And the most important thing about all this is that every single one of these data sets comes from a completely different database. And many of these are internal, many of these are external. Almost all of them are not on this slide. And the overall scope of the project is to get them all in one big pot, construct a giant interaction diagram of the ways these things interact, and then figure out what we can from that. Also, the scope is much larger than this over the next few years. So right now, genomic information is the primary thing. We're also wanting to put in compound information, perhaps imaging information, protein interactions, and other kinds of things that we want to feed into this graph over time. And another thing that's particularly hard in our scope is we need to be extremely flexible. So a lot of our data comes from completely different sources we have no control over. They think about the data in different ways that we might not agree with, but we have to model that because that's how they did. And also, even within Novartis, our own scientists have disagreements among themselves about how to best model the data for different kinds of problems. And we have to be flexible to accommodate all of their needs. And another way we need to be flexible is in terms of analytic technologies. So different scientists have very different use cases and very different kinds of tools that they're used to using. So we need to be able to take data from our graph and put it into these different tools relatively seamlessly. So that's just sort of the problem that we're trying to solve. In terms of the, I guess, the characteristics of the data that we're dealing with, currently in our graph we have in the low trillions of edges right now. And when we flatten it out into tables for one particular schema, it's in the hundreds of billions of rows. And that's not, I guess, maybe so vast in the big data space, but it's reasonable and it's definitely not tiny. But it's also growing very fast. So most of our data comes from these things called sequencing machines, where you put in a piece of tissue and then out streams a huge amount of data. And the first time people started doing this around 15 years ago, it cost about $100 million every time you wanted to do this. And so they didn't do it much. And now the price is down, it's falling exponentially. We're down to like $1,000 to $2,000 every time you want to do this, and we're still falling. So the prediction in the future, and just in the next 2 or 3 years, is that we're going to be swimming in this data even more, and we're looking at exponential explosion even over these numbers. So we're trying to plan for that. Another thing about the data is that it's ridiculously messy. Part of that is because it just comes from so many sources, internal and external. And part of that is it's not totally clear that our external partners actually have an incentive in providing the data in a standardized form. So there's more to talk about there. But that's basically the problem space. The overall architecture of this is hopefully not quite as complicated as the picture seems, but the main thing to say about it is that the system is a framework more than it is a single database. So the basic idea is it all runs in batch mode, and we take a lot of data, we bring it into this one big giant graph, and then we run analyses on that graph, but then we also spin out subsets of that graph into different analytic databases for different kinds of purposes, maybe to drive a GUI, things like that. And it all begins with, just to sort of walk through this, we have data on the left side, and that might come in CSVs or any sort of other standard format here. And the first thing we do with this data is add what we call markup. And markup is kind of an important thing for us. It's a Scala-based domain-specific language. And what it is for every particular data input file, we have an associated markup file. And that markup file tells us what is in that data file, but it also tells us how to integrate the contents of that file with the rest of the giant graph. So where to draw the edges so that it appropriately links in. And once you add the data and the markup files together, you get our giant graph. And all of the production of this graph occurs in Spark. We're running all this in our in-house Hadoop cluster. So once this is done, the graph is persisted out into HDFS. But like I said, all the actual mechanics are in Spark. Then from that graph, there's what we do on the output side, what we do to actually get at the data. And there's two main ways we do this. First, we have what we call an analytics API, which is basically a set of libraries that people can use if they're trying to run a Spark job that can get at the edges of the graph, and then they can do whatever they want with those edges. And then the second way that people get at the data is we have this thing called table markup, which is another domain-specific language. And the aim of table markup is it's targeted more at scientists, is for them to be able to express how to pull out a specific piece of the graph into a relational schema or a set of CSVs of a particular schema, which then they can load into their tool of choice. And that's basically our answer for providing sort of Spark-based analytics and also all these other legacy tools that people are familiar with. That is the overall picture. I'm going to say just a tad bit more about the way we actually generate the graph. The most interesting thing about it, perhaps, is that after we parse the data, we split it up into what we call, these things we call semantic units. And that is like if we have a data set of mutations coming in, then each mutation is one semantic unit. If it's a data set of diseases coming in, then each disease is one semantic unit. And as you bring this stuff in, the first thing we do is that semantic unit will have a list of instructions which says how to interpret for the given lines of original data file, how to interpret that as edges with respect to the rest of the graph. And then we do a lot of Spark manipulations to actually create the edges. And at that point, we have what we call kind of a first cut at the graph. After we have that first cut, we do various pieces of logical enrichment to make we add more edges to it. We basically do things like type inference and some other custom logic in order to enrich the data so that it's more suitable for processing downstream. After that, we write it out, and it's good to go for the next few layers. Some questions that we often get are why graphs? Like, why do we do all of this with graphs? There's 2 or 3 different answers to this. One is that we really just wanted to represent all of the data in terms of binary relationships. So something like this tissue has a diagnosis of adenoma, or this gene is at this location. We like representing things that way as our fundamental data structure for a couple of reasons. One is that it's a lot more flexible than tables. So you write out a schema in a table, you can't really modify it much. And one of our driving requirements is that we must be able to modify this and do different things frequently. And at the same time, these are more expressive than key values. So key values are pretty flexible, but it's pretty hard to describe an extremely complex system just with a set of key values. And another reason we're using these is that binary relationships support, there's a huge literature about doing logic on these things. You can do transitive closures, subsumptive reasoning. It's all pretty easy. And that's very useful for us for validation purposes and for integration purposes. And another reason is that binary relationships are just semantically the exact same thing as a set of labeled edges in a graph. So it's very easy to go from this fundamental data structure into a graph, which means that different kinds of topological or network analyses on the graph are pretty easy to get to. And we would do those with something like GraphX. Which leads to a separate question, is why are we not actually using GraphX for any of this? And the main answer is that things like GraphX, the graph toolkits, typically think in terms of vertices and edges as their fundamental structure, whereas we tend to think of these semantic units as our fundamental structure, which might include some hundreds or maybe thousands of edges in each one. And the reason we do this is that for semantic units, there's a very natural mapping between maybe like 5 lines of input file to a set of instructions for interpreting that in the context of our graph, and then a set of edges, and then we can even show which rows that semantic unit will contribute to other downstream databases. So in terms of a semantic unit, we can see a whole window of the integration process from where it began and the raw data files to all the different transformations and some of the reasoning behind those transformations down to what rows it will contribute to individual databases. And this is very natural to do in Spark. It works fairly well. It's kind of awkward for GraphX. But the great thing is that GraphX is just a flat map away for whenever we actually need to use it. The future of this is, as I said earlier, it's not just genomic data. That's our focus now because it's pretty large and it's pretty important for us. But we're definitely building in, and we have interest in modeling all kinds of other information in this. And also in our future is more things from the Spark ecosystem here. So Parquet, data frames, likely Atom. And if you don't know anything about Atom, you should attend Timothy Danford's talk at 5:30. And also, future is just bigger, probably. So we're around 1 trillion edges now, a bit over, and we're likely following this exponential trajectory over the next few years. So I just want to acknowledge the people in red are people who are on our team or now or at one point in time, and people in black who are not formally on the team but have helped us out wonderfully at one time or another. And also want to add that we're hiring very much. So if you're interested in Spark and you're interested in trying to help make new medicines, we have this great campus right beside MIT in Cambridge. And please apply. Or if you're from Europe, we also have a lab in Basel, Switzerland, which is very nice as well. And that's more or less it. So please ask questions or catch me later.
OK, any questions? We can cheer for him, too, but we also want to ask questions.
How many sequenced people does that represent in the 4 trillion there?
I'm not sure I'm allowed to say that. But if you looked at all the large public data sets and genomic data that are out there, that'd be kind of about right. That'd be order of magnitude.
More questions?
I'm sorry if I missed it, but can you give an example? Because you have a very generic framework, but how do people ask questions to the generic framework, given the fact that you sort of have disease data and DNA data and protein data all in there?
Yeah, so that's a great question. So the 2 ways of getting at it, the direct run on Spark Analytics API is pretty much centered on power users. You get the edges directly. You have to know how to manipulate those edges and what data sets they come from. And it is very much, there's not many people who are capable of, who know the system well enough to do that. One of the endpoints is a relational database, and so we spin it out to there, and then we have some R APIs that are just sort of clean APIs for hitting that, and those are going to evolve over time. But the idea is that that's our user-friendly kind of piece.
Yeah, from what you've explained, you have basically 2 selector framework in which you ask for the correlation between one phenotypic kind of data and one very genotypic one. How do you see that evolving in the future? Do you see that to be a constant of your model forever and ever? Or if not, how would you go about evolving that?
Do you mind repeating the question?
Oh, yeah, sure. So the question is, as I understand it, is it seems like we're focusing largely on correlations between like a phenotype and a genotype here, and how is that likely to evolve over time? That is certainly our main use case, is that kind of stuff. I'm not entirely clear if that's maybe even half of it right now, just because there's so much information about clinical metadata in there about the samples. And probably the real answer to your question will change depending on the different kinds of data that come in. So compound data is likely coming in, and there the questions are more along the lines of, does this compound change expression values at this location or something like that? So on a sky's the limit. It's basically, we're trying to solve the bring the data together problem sort of first, and then hopefully better questions come out after time.
Okay, we have time for one more question if there is one. All right, thanks a lot.